Hello, everyone, and welcome to today's webinar. My name is Rula Deeb. I'm a senior principal at Geosynta Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. Today's webinar will consist of a brief overview of both programs, followed by technical presentations on DoD-funded research on 1,4-dioxane biological approaches. First, Dr. Shelly Mahendra from the University of California, Los Angeles, will discuss the use of a microbial co-culture to rapidly biodegrade mixtures of both 1,4-dioxane and chlorinated volatile organic compounds. Shelly's presentation will be followed by a brief Q&A session. Then Mr. David Lippincott from Aptum Federal Services will talk about the implementation of an in-situ co-metabolic barrier to treat large dilute CVOC plumes. Today's presentation will also be followed by a Q&A session, and we will conclude the webinar with a longer Q&A session featuring both of today's speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and provided to you in your webinar registration confirmation email. If you're unable to download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you have difficulties viewing the slides or if your screen freezes, try keying in Control and F5 to perform a hard refresh. If you are accessing the audio through the computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties with the audio, feel free to call into the conference line shown here and provided to you in your registration confirmation email. You can also submit a comment using the chat box. Please use the chat box to communicate with us regarding technical difficulties. Do not use the chat box for submitting questions for the speakers. The Q&A option should be reserved for questions for the speakers. In case of continued difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration email. Note that we will be live streaming the webinar on the SERDP ESTCP YouTube channel, and that's another option for following today's proceedings. The broadcast today will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. We, in fact, encourage you to submit your questions in advance of the Q&A sessions. We ask that when you do submit your questions, to make sure that you add your organization name at the end of your question, and we will do our best to identify you during the Q&A session. With that, I would like to introduce Dr. Andrea Luen, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Dr. Luen has been with SERDP and ESTCP since 2001, and prior to that, she served as a research leader at the Patel Memorial Institute, where she conducted scientific research on in-situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed.
Great, thank you, Rula, and thank you to everyone who's joined us today. I wanted to just cover a very little bit about who we are in terms of SERDP and ESTCP before I turn you over to our presentations today from our guest speakers. And I'll talk about our two companion programs. The first is SERDP, the Strategic Environmental Research and Development Program, and this is our, what you would consider a traditional science and technology program. We have been around since 1991, and we are a partnership between the Department of Defense, the Department of Energy, and the Environmental Protection Agency. And our goal really is to fund fundamental research that will help us address our real-world environmental management issues. Our companion program, ESTCP, the Environmental Security Technology Certification Program, now this is a demonstration validation program. And under this one, we take the research or the knowledge that comes out of SERDP or other research programs, and we take it to the field and look to validate that so that we have advanced the research to the point where an end user can now take it and adopt it at their sites. And so we're really looking for this direct technology insertion. And as you can imagine, this technology transfer is very important in this process. We have a couple of main drivers that guide the sort of research that we fund. The first is the sustainment of our testing and training ranges, the facilities and operations on our installations. You can imagine this is very broad. It takes the form of addressing unexploded ordnance and the associated munitions constituents. We'll address toxic air emissions and dust, threatened and endangered species. So it's just tremendously broad number of issues that we're concerned with. Likewise, we're also interested in reducing the current and future liability on our installations. That could take the form of the really traditional program of pollution prevention, where we're looking to eliminate or replace hazardous pollutants and hazardous processes on our installations. And we're also looking for achieving compliance through this pollution prevention. And then we also deal with contamination from past practices, and the presentations you'll see today are really addressing this issue. That can include impact to our groundwater, soil sediments, our UXO liability falls under here, as well as any kind of emerging contaminants that we learn about as we go along. And so this really drives some of the research that we fund. Always important to us then, though, is to transition our results out to end users and to the broader community. We do this through a number of means, and you can find all of this on our website listed here. You can find videos. We have so many guidances and manuals. We have an EnviroWiki, similar to, of course, the Wikipedia. We do in-person training, and then, of course, we do these webinars. So these are a key part of our technology transition process. Listed here are just a few of the webinars that are coming up this year. We have webinars roughly about twice a month. And if you go to our webinar page, I will show you that link in a moment, you can register for these webinars, and you can see which ones really suit your interests. So as I said, you can go to this page, you can find everything there is to know about upcoming webinars, you can register there. You can also get the archived versions of webinars there as well. So if you've missed one, you can find it on the website and listen to it at your own leisure. So at this point, I'll turn it back to Rula to introduce our next speaker.
Thank you so much, Andrea. It is now my pleasure to introduce Dr. Shelly Mahendra, who is a professor in the Department of Civil and Environmental Engineering at the University of California in Los Angeles. Shelly has served as a principal and co-principal investigators of projects funded by SERDP, the Air Force Civil Engineering Center, the National Defense Center for Energy and Environment, the Naval Facilities Engineering System Command, and various other organizations. Her work has focused on a range of emerging and established contaminants, including 1,4-dioxane, chlorinated solvents, PFAS, nanoparticles, pesticides, and munitions constituents. She has received multiple national awards in recognition of not just her research, but her teaching and professional activities. Shelly has authored more than 70 peer-reviewed papers and book chapters, and she received her doctoral degree from the University of California at Berkeley. Go Bears. Shelly, we're happy to have you. Please get us started with today's first technical presentation.
Thank you, Rula. And thank you, Andrea and SERDP ESTCP, for giving me the opportunity to share our research. This is from a limited scope SERDP project in which I describe how aerobic and anaerobic bacteria can team up to biodegrade CVOCs and 1,4-dioxane. To give you a roadmap of what I'll be talking about, the problem of contaminant mixtures, technical objectives in my project, and how we selected and formulated and synthesized and made it work on seemingly incompatible aerobic and anaerobic microbial communities. And since we are talking about aeration, the dosing and the timing of oxygen becomes really important for the growth and biodegradation processes by these bacteria. And not only do we show work that was limited to the lab, but some amount of in-well biotrap studies to get a sense of how this would work in the environment. And I'll end with conclusions and how some of this work benefits the DoD and stakeholders. Many of us know contaminant plumes are contaminated with mixtures of substances. In case of chlorinated solvents, some of these solvents use dioxane and as a solvent stabilizer. As a result, different solvents were used over time, different stabilizers. What remains in the groundwater are plumes, which contain various zones of contaminant mixtures, different lengths and coverage. That's not important. What we found in a previous SERDP project with GSI was nearly 95% of the dioxane plumes contain one or more chlorinated solvents. So anything that has mixtures, if we want to remediate them, we want a technology that can take care of diverse contaminants, preferably at one shot. Speaking of biological degradation, luckily 1,4-dioxane can be biodegraded by bacteria that can use it, it being dioxane as its sole source of carbon and energy. So it can grow on it as food. One such microbe that I worked with for 15 some years is Pseudonocardia dioxane strain CB1190. And the really cool thing about this organism or metabolism is we start with 1,4-dioxane and it's completely mineralized to CO2 relatively rapidly too, and importantly, without accumulation of toxic intermediates. But this process is aerobic. These bacteria respire oxygen as terminal electron acceptors. Chlorinated ethenes are at least initially degraded anaerobically for highly chlorinated compounds such as PCE or TCE. There's some reports of other lower chlorinated ethenes being degraded aerobically, but in general, TCE and others. And that process can also take place biologically. There are organisms dehalogenating or reductively dehalogenating organisms that are strict anaerobes, are very well described and established, successfully degrading, reducing TCE to ethene. And so we know this stuff is not new in my project. We knew there are aerobic dioxane degrading bacteria and anaerobic chlorinated solvent degrading bacteria. Our goal was how to get these two, opposing processes, opposing redox conditions, and we want this to happen at the same time and get these, enemies become friends and inhibitors become co-substrates and oxygen adaptation works out. Those were what were the goals of our project. It was a little bit of ambitious project, limited scope, but I'm happy to report that will show some really exciting data. Technical objectives were to design and test the microbial communities that would achieve simultaneous or sequential degradation of CVOCs and dioxane. So under certain anaerobic conditions, part of the process would take place, things become aerobic, everything gets mineralized to CO2. That's the plan. And understand the influence of oxygen dose and timing on the kinetics and overall mass removal. And back it up, all the observed degradations and rates by measuring taxonomic abundance and biomarker gene abundance and expression, so that we know there's a molecular foundation of what we are observing in terms of quantitative data. So let's dive into the results. What I'm showing you here is time in days on the x-axis and concentrations of chlorinated ethenes in micrograms per liter on the y-axis. The gray shaded region towards the left side indicates anaerobic phase, and that was selected up until TCE was degraded to below detection. And that's what's shown in the solid green line. That is TCE. We have a single bottle in which KB1, a reductively dechlorinating culture that was generously provided to us by CIREM, is mixed together with aerobic CB1190 in a secret sauce, but both organisms, groups were present at the same time, and it started with anaerobic, and then we changed the conditions to aerobic after TCE was all gone. And when TCE was removed, the dashed green line that indicates cis-TCE appearing, that was formed. Under aerobic conditions, cis-TCE declined. There was no vinyl chloride detected. And that happened in the mixed communities. On the secondary y-axis, you see abundance of KB1 in copies of TCA genes per mil. And TCE genes, they increased. So the column graphs that you see at the bottom under anaerobic phase, but not much change. In fact, there was some decline on exposure to aerobic conditions. Similar plot, but now we have on the y-axis is 1,4-dioxane. Not much dioxane degradation occurs under anaerobic conditions. And remember, KB1 and CB1190 were added together. When conditions become aerobic, dioxane is degraded. The abundance of CB1190, which is indicated by these columns, doesn't change much under anaerobic conditions, as you would expect, but increases as the cell population grows while degrading dioxane aerobically. So this is working really well. So initial anaerobic TCE reduction. Once things become aerobic, cis-TCE, dioxane, all the other things are removed aerobically. It also incidentally happens to be first report that CB1190 can biodegrade cis-TCE. And it's in somewhat contrast to previous work reported from my own lab, also funded by SERDP, in which cis-TCE was considered an inhibitor of dioxane degradation by CB1190. We now think that the mechanism is a little more nuanced. It could be somewhat inhibition, but also competition for the monooxygenase enzyme. And whether dioxane is present or not, cis-TCE and dioxane can be simultaneously degraded by the pure culture of CB1190. And that's a result that we are sharing. Since we're talking about dioxane, other concerns appear that CB1190 strain uses dioxane as a growth supporting substrate. So like every other microbe, it will have a minimum substrate concentration at which the growth is viable. What we are showing here is CB1190 can sustain growth and degradation of dioxane at concentrations as low as 10 micrograms per liter. So it's different from what previous work also conducted under SERDP has shown. But we understand more about the microorganism now and the conditions under which we have updated kinetics, Vmaxes and Kms, and 10 ppm, 50 ppm is much more respectable. Higher concentrations are obviously fine. Can go up to really, really high concentrations, 100,000 ppm would also be good. And so in the light blue triangles over here, we do show that 10 micrograms per liter dioxanes happily degraded by CB1190, and this flat line just indicates the detection limit of my GC mass spec at that time, but I suspect it could go even lower, starting at 10. So that was the story with electron donor, dioxane. Let's talk about electron acceptor. So CB1190 is an aerobic, more accurately microaerophilic organism. It can degrade dioxane with as little as about 3 milligram per liter oxygen. And that is following prolonged anaerobic periods, even in the presence of low concentrations of dioxane. This specific plot shows high concentration, 50 ppm, but in the presence of solvents, anaerobic incubation, all kinds of things. So you don't need saturation, you don't need high 7 or more milligram per liter dissolved oxygen to achieve aerobic dioxane degradation by CB1190. So let the fun start. So here, longer term experiments, time in days on the x-axis, dioxane concentration on the y-axis, and these gray dashed lines that you're seeing, that's dioxane concentrations. Dioxane is degraded, we add more. Conditions are always aerobic. This serves as our positive control. We also have a negative control, that's the dotted flat line. No dioxane is degraded in under anaerobic conditions. But the blue curve, so what we are showing here is over 1 week of anaerobic incubation, when oxygen is added, dioxane is rapidly degraded the subsequent weeks. So earlier it was a matter of hours of anaerobic period in which TCE was degraded. Now we are keeping anaerobic conditions up to 7 days, 14 days, 2 weeks, 21 days, 3 weeks. Notice no degradation under anaerobic conditions. As soon as oxygen becomes available, it's almost like the microbes were ready to launch. Very minimal lag. The degradation rates, the slopes are about the same, if not better, after anaerobic incubations. 4 weeks, 5 weeks. So up to 5 weeks of anaerobic incubation, oxygen slugs added, dioxanes happily degraded to below detection. And the biomarker data corroborates what we are seeing here. Over time, if you look at CB1190 abundance, the blue and the orange bars indicate the anaerobic phase. Not much change happens there. That's just like you would expect. The green columns are abundance of CB1190 after it had a chance to degrade dioxane aerobically. And that shows increase in population. So the cells were viable during the anaerobic phase. They just didn't grow. When you look at gene expression, exactly what you would expect. The gene coding for dioxane monooxygenase, a component of that, DXMB, and ALDH is gene that codes for aldehyde dehydrogenase. That degrades products of dioxane degradation. So we know that the pathway is sustainable. It will result in mineralization downstream. Together, they serve as very accurate and confirmatory biomarkers for dioxane biodegradation. And over 56 days, exactly what you would expect with respect to these quantities. And some of these data are reported in an open access editorial choice paper, Environmental Science and Technology Letters, 2019. Also included in the SERDP limited project report that should be posted on SERDP's website. So 35 days is good. How long can CB1190 survive? You know, we are aerobic organisms as humans. We can hold our breath for a little bit, but really, how long could you go without oxygen? We tried and we found 100 days was fine. 200 days of anaerobic incubation, we added oxygen, dioxane was still degraded. So there was not much growth of the organism, no degradation, but the organism did not die off. CB1190 hung in there, waiting for oxygen to appear so it could start degrading dioxane and get fat on it. And that data I've shown so far indicates when there's a period of anaerobic phase, however long, and then followed once by aerobic conditions. In a real aquifer, variabilities, heterogeneities occur. There are pockets of aerobic and anaerobic. Things can go back and forth with changing water chemistry. There could be a pump during biosparging that could break down. What we are envisioning is, now that we've figured out how to get KB1 and CB1190 to work together and grow together, in a real injection, you could just add them together at once. And then KB1 would degrade TCE up until cis DCE, And then as oxygen becomes available or is injected, CB1190 can take care of residual cis-DCE and other things, as well as dioxane when conditions are aerobic. Of course, KB1 would not survive exposure to oxygen. Next few plots would be similar to what I showed earlier. Time in days on the x-axis, ppbs of dioxane, and those gray shaded regions are anaerobic phase. The orange curve here shows aerobic always condition. So add dioxane, CB1190 degrades it right away. If you hold it for 1 week anaerobic, it'll stop there. In the anaerobic condition, it'll degrade again. Add more dioxane, cut off oxygen, nothing happens, things are standing still. Add oxygen, 1 week later, dioxane is all gone. 1 week anaerobic, 1 week aerobic. Let's do this. 2 week anaerobic, 1 week aerobic, and those are the purple curves. So they hang in there, aerobically dioxane's degraded, but back and forth, there's no change in slope, there's no lag, there's no inhibition. 3 weeks anaerobic, 1 week aerobic, same story. Dioxane's not degraded anaerobically, but the culture is viable to go across back and forth, toggling between aerobic and anaerobic conditions. And the same plot without the shaded region, so you can see the slopes. And as I said, the cells are viable, usually going back and forth between aerobic and anaerobic in incubations, and solvents are present and limited oxygens being added and stuff's going on. You think some of the population could decline, but what we are showing in biomarker, DXMB or even 16S ribosomal RNA, taxonomic genes would show the same thing, that there was no change in population, there's no decline in CB1190's abundance after these 1, 2, 3 weeks toggle of aerobic, anaerobic fluctuations. And even though it was a limited scope project, we wanted to really have this proof of concept that this could work, getting aerobic and anaerobic KB1, CB1190 cultures to grow together, degrade chlorinated solvents and dioxane all at once, degrade cis-DCE, back and forth, all that. But we wanted to have a foot in the real world to show that this can actually work in the field. So we collaborated with Dr. Laurie Lepat Velasco of Matrix New World Engineering. She was working at this site which had been subjected to enhanced reductive dechlorination with SDC9 culture for about 2 years off and on. And then we grew CB1190 on biotraps, microbial insights, biosep beads, biotraps, and suspended them in monitoring wells. So the source is somewhere here in the bottom left corner. Groundwater flow direction is indicated. The first well, MW30, because the source was recently clean, it actually had relatively low dioxane and CVOCs. And MW31, down gradient from there, had high dioxane and CVOCs. What the data show is over 120 days, 4 months or so, remember, CB1190 can survive 100 days or more of anaerobic incubation. Dioxane, which is indicated in these light pink lilac triangles here, started around 3,000 some micrograms per liter. And after 4 months of being suspended, just 2 monitoring wells in that entire site that had long-term anaerobic conditions to promote reductive dechlorination. There was from 3,000 something, dioxane degraded to about 1,300 micrograms per liter. At the same time, there's not much change or slightly increase in TCE concentrations. The data on the bottom plot shows biomarker gene abundance, copies per bead. These numbers are a lot more reasonable than some of the lab copy number levels, the really high density cultures we were trying. Basically, what we are showing here is that the organisms that express DXMB and ALDH, dioxane degrading genes, can occur simultaneously coexist with TCEA and VCRA, so anaerobic organisms, in this aquifer in a real site, not just in the lab. The other well where I said the concentrations were lower, we see the same behavior. Dioxanes degraded. TCE and cis-TCE, not so much. In more recent data than what I'm showing you here, we do see decline of cis-TCE also. And copy numbers again, aerobic and anaerobic gene containing organisms can coexist. So just let me remind you what I just described. It was a one-year project. We assembled a microbial community that is able to survive changing redox conditions and efficiently degrade CVOCs and dioxane. So that's a KB1 CB1190 combo. So CB1190, now we know aerobically degrades cis-TCE, and there's no chance of forming vinyl chloride because the reaction's aerobic. Cis-TCE, as does dioxane, get mineralized to carbon dioxide. CB1190 can also withstand over 100 days of anaerobic incubation and just be ready to launch when conditions become aerobic and dioxane will get degraded. And you don't need a lot of oxygen, just about 3 milligram per liter is good enough to sort of sustain dioxane biodegradation. And this works in the lab, and we are continuing to understand and characterize and fine tune and tweak using advanced omics tools to understand what's going on, but at the same time also demonstrating that the CB1190 culture can remain viable and active even when sites were previously anaerobic. Some of this work we think would be beneficial to many of you, the Department of Defense, our friends in the practitioner communities, other academic researchers, regulators, that the CVOC dioxane degrading communities were formulated, and they could serve as a model for other contaminant mixtures. But in this case, I think the benefits were that we know that these organisms can now be trained to work together, and we bypass the formation, accumulation of toxic intermediates like vinyl chloride, which means that a single injection should be good enough to take care of CVOCs and dioxane. You may need to limit the ERD timeframe, just take care of TCE depletion because cis and other CVOCs along with dioxane can be degraded by CB1190 under aerobic conditions. So this would be very valuable at some CVOC sites. And as I said, the cost might be lower than comparable physical chemical processes, of course, but also biological because you need fewer injections of microbes. This is not a co-metabolic process. You don't need to add primary substrates like propane, and even limited nutrient amendments and oxygen amendments are required. From the science point of view, I think this project, like SERDP's research project, will help us understand how environmentally relevant microbes adapt to different biogeochemical conditions, and that's important to improve remediation and monitoring activities. And we also can use these data and findings from this project to design and select biostimulation strategies, cultures, bioaugmentation, their doses, their ratios of KB1, CB1190, understand what could happen under fluctuating dissolved oxygen levels, and low concentration of contaminant mixtures, as well as near source zones, high concentration. So just working throughout all those things, I think results from this project would be really beneficial to the DoD and others. Just a list of papers that have been cited. Some are by us and by original authors to whom I want to provide credit. And this last paper was directly a result of our project. So that's available, open access. I just want to thank everyone for the attention. Thanks, SERDP ESTCP again. And I'm happy to have a discussion, fill in some gaps, clarify anything you might have, and answer questions. Thank you very much.
Yes, thank you, Shelly. As a reminder for our audience, you may submit questions by using the Q&A box on your screen. We have received several questions that Shelly will now answer, starting with a question from colleagues at Geosyntec. The density of microorganisms in test cells seems quite high. What are typical levels achievable in the field? Also, do you have kinetic parameters for this culture?
Thank you, Mike. In our early experiments, we wanted to be sure that it works because it was, in some ways, a little high risk, high reward kind of a project. So early experiments did use really high cell densities, 10 to the 10, 10 to the 11 cells per mil. In subsequent studies, we have shown that even lower 10 to the 5 cells per mil are enough to achieve dioxane and lower chlorinated solvent biodegradation. We do have kinetic parameters. We're fine-tuning them as part of ongoing work under SERDP for this project to get affinity constants and inhibition constants and accurate rates. But overall, it's very, very promising. I could look up and tell you exact numbers in a little bit, but yeah, in our first attempt, we wanted to make sure that we give it every opportunity. As we thought some of the microbes would die, so we started really high. But in subsequent studies, we didn't need as much. So several orders of magnitude lower were enough. I would say 10 to the 5 cells per mil is good enough for lab and field studies.
Great, thank you so much, Shelly. Very exciting results. This next question is from one of your ex-students who's now at GSI Environmental, and he's asking about some of the possible reasons that CV1190 can survive for so long under anaerobic conditions.
It's always nice when my students point out, you know, in one way we train our students, and the thought process is the same. That's the question we've been asking ourselves. What happens is that this organism is very versatile in its metabolism. It has various ways, it's even an autotrophic organism, and it can subsist under different mechanisms under anaerobic conditions. It just won't degrade dioxane because dioxane is degraded by monooxidase enzymes, which has a requirement for oxygen present as a terminal electron acceptor. But various other cellular processes allow the microbe to just have low levels of substance, just not dioxane. It's probably fermenting and doing other biochemical processes to stay alive. And we do discuss that. There are other ways in which low levels of reductants can keep the microbe alive for a period of time under anaerobic conditions. We can all hold our breath. We are aerobic organisms, but how long can you hold your breath? That's what we wanted to test for this microbe, and it came up really surprisingly nice for the number of days. So yes, there's some early thoughts on why anaerobic subsistence is reasonable for this microbe. Our data are not artifacts, those are real. But work is ongoing to really get into the omics and real fundamentals of what's happening in the cell.
Thank you so much. And a follow-up question to this, Shelly. Do you have thoughts as to why 1,4 biodegradation resumes so rapidly after the introduction or reintroduction of dissolved oxygen?
No, I'm as surprised as everyone else. I do think that the genes for dioxane degradation are induced by dioxane itself. The enzyme, when the genes are transcribed and translated, the enzymes form, it's ready. The enzyme won't work until oxygen's available, but the enzyme is produced and ready to launch as a result of dioxane exposure and some other inducing substrates. So oxygen's required to kickstart, but to produce the enzyme, you need dioxane. So any site that has dioxane, even like we are showing at low concentrations, low ppbs, would keep the enzyme synthesized and ready to degrade dioxane when oxygen becomes available. And I was very, happily surprised because when we sequenced the genome of this organism, CB1190, the genes for dioxane degradation are actually found on the plasmid. My bigger concern at the beginning of this project was that in prolonged anaerobic conditions, the microbe would just drop the plasmid, and that did not happen. So yeah, we're very happy with the results. Again, to summarize, the production of the enzyme depends on dioxane. The activity is triggered by the presence of oxygen.
Thank you, Shelly. We received 2 questions about how low can you go with 1,4-dioxane from Terra Systems and Langen. So is there any way to biodegrade low levels of 1,4-dioxane when you get to, let's say, the 10 microgram per liter levels?
Well, and I get asked this question all the time. In my early work, 15 years ago, I think we had analytical limitations. We know more now than we did then. So the published rates of affinity constant, half saturation constant, tend to be in the milligram per liter, but our recent work repeatedly shows that as low as 10 or 50 micrograms per liter, possibly even lower, can be supportive. And the reason for that is earlier we were looking at simple systems, and that makes sense. Our training, we learned to walk before we run. We only had one organism in a limited medium, and dioxane's present. What I think could happen is that in a real environment, in this case, the field may provide complexity, which is harder, but it could also work to our advantage, that other substrates present can keep the microbe alive, just create the enzymes, the CCE, who knows? That might be something that is helping the degradation of low dioxane levels, low micrograms per liter dioxane levels. Right now, because the detection limit of my GC mass spec was around 4 or 5 micrograms per liter, that's the lowest level I could confirm, but I suspect it could go lower. And it's not the fault of the microorganism. There is a minimum concentration that supports the growth and metabolism. It's the degradation at low levels or any chemical reaction, which is really just a game of probability, is hard at very dilute concentrations. But I do think it can happen. The rates will be lower, but even low tens of ppb of dioxane can be aerobically degraded by bacteria such as CB1190. Maybe there are other ones out there. In my studies of CB1190, it's very encouraging.
Thank you so much. Let's shift gears to next steps, Shelly. We have a question from TU Dresden in Germany. If you were to apply this, your proposed technology in a field setting versus in the lab, do you believe that the treatment system will be sensitive to field conditions? And specifically, the question here was targeted to temperature. Have you found CB1190 to have any temperature sensitivities?
We are trying, now that we are encouraged by our results obtained so far, we are trying to push it further, how various environmental biogeochemical conditions under which this would be viable. The pH tolerance, this is a circumneutral physiologically adapted organism. So I think the lowest I have measured is around 5.5. It did fine. I have not tested at very low or very high pH levels. But there may be other, ionic strength. This culture in my really previous studies, doctoral studies, it was able to tolerate up to 4% salt, stress, other considerations. And it's always nice when my own students prove me wrong, whether it's dioxane absorption, whether it's anaerobic survival of CB1190, whether it's showing that chlorinated solvents can be inhibitory to dioxane degradation, and we quantified that and characterized that. And then we turned around and said, by the way, CV1190 can actually degrade some of those potential inhibitors, so it's kind of in a circle. Overall, it's better. And what we thought was inhibition might have been competition. And just like Mike or somebody else asked the question about the kinetics of Vmaxes and Kms and inhibition coefficients, once we have a better picture of those data, we'll be able to predict much better. The short answer is it depends, but at least in the things that I have tested, dioxane concentrations, chlorinated solvents concentrations, oxygen levels, pH, and a few other things, in many sites, more sites than I had previously imagined, whether it's a bioaugmentation with CV1190 pure culture or together with KB1. I think it's more sites than we would have thought where this is likely to work.
Thank you, Shelly. One more question, and we promise to come back and answer the remaining questions after we finish the second talk, but this is from Golder Associates. Any thoughts on introducing CV1190 into the vadose zone along with oxygen to create a biobarrier to prevent horizontal migration? Would pore water be enough to sustain the culture?
Thank you for the question, Bill. Our thoughts exactly. We are in conversation with a group where we would want to introduce CV1190 in a biobarrier. Some of the early work in my lab we looked at, inspired by Jim Spain's work, looked at CV1190 growth and possible biodegradation of dioxane in the capillary zone. So even some vadose zone studies would be valuable. I don't have the data right now, but we have in-situ reactors columns underway and plans for testing the biobarriers in the next few months. So stay tuned. But I'm glad to
thank you.
Start thinking in the same direction. So it gives me validation.
Thank you so much, Shelly. Great responses to all these questions. And again, we'll get back to the remaining questions towards the end. But it is now time to introduce our second speaker, Mr. Dave Lippincott, who is a senior project geologist and researcher in the Biotechnology Development and Applications Group at Aptum in New Jersey. Dave's current areas of research include the development of in-situ technologies for recalcitrant and emerging groundwater contaminants, including chlorinated solvents, PFAS, 1,2-dibromo, methane, ethane, NDMA, and 1,4-dioxane. Dave has authored several papers on the successful field applications that are associated with the biological and non-biological treatment of these types of compounds. He earned bachelor's degrees in geology and business from Stockton University in New Jersey. Dave, please proceed.
Thank you, Rula, for the introduction. I'd like to thank Andrea Luen and the SERDP ESTCP program for giving me the opportunity to talk about this ESTCP project, where we evaluated a co-metabolic barrier as a sustainable approach to treat a large dilute CVOC foam. The agenda for my talk today is we'll start out with a brief discussion on the large dilute plume problem and overall objectives for this demonstration. Talk briefly about some background on co-metabolism. I'll spend most of the time on the ESTCP biobarrier field demonstration project, the various different aspects of that. And then at the end, talk about the conclusions from this project and how this could potentially benefit the DoD and other organizations. So large dilute plumes are, generally a problem because of their size and because of trying to achieve regulatory levels on such a scale is often impractical or cost prohibitive. And one of these reasons is even if you do treat the permeable zones, you'll definitely get back diffusion over time from the contaminants that have diffused into lower permeability layers, both in the source area and downgrading of the source area over the years that the plume is persisting. So these plumes are typically sustained for decades or beyond because of these matrix diffusion and mass transfer processes. And also if you have slow natural attenuation processes, be they abiotic or biological processes, these plumes can last a long time or even expand over time. So the technical objectives for this project were to basically look at a co-metabolic treatment approach for these large dilute CVOC plumes. Something that would be sustainable and semi-passive and cost-effective, but also achieve regulatory limits, in this case, the MCLs for cis-DCE and vinyl chloride. We just have a cartoon on the bottom showing the co-metabolic biobarrier approach and how we envisioned it. So just a little brief background on co-metabolism. So Shelly spoke about the CB1190, and that is, if you look at the cartoon on the left, that's basically a growth length biodegradation process for Dioxane. Here we show benzene, where these cells, these bacteria, they gain energy and carbon from the contaminant itself. So they're actually growing on the contaminant. Higher concentrations of contaminant can mean higher growth for these organisms. But when you have low contaminant concentrations or conditions that aren't good for growth, like in this case, a lack of oxygen, you're not going to have very good kinetics associated with the degradation via this process. So if you look at on the right, the co-metabolic biodegradation process, these cells do not gain any energy or carbon from degrading these contaminants. Actually, they won't degrade these contaminants until you add another substrate, a primary substrate such as propane or methane or something similar to that can get these oxygenases expressed in these organisms, and then they're able to degrade these different contaminants. And a lot of these oxygenases have the ability to degrade several different contaminants. These organisms can express several different oxygenases and have the capability again to degrade a mixed contaminant plume. So what are some of the advantages of the co-metabolic approach? Well, as I discussed, we're adding a primary substrate, and these organisms are growing on that supplied substrate. So instead of relying on the low concentrations in these dilute plumes, they're actually growing on this propane or whatever other substrate we determine is best that we're adding to the aquifer. And you can get really high biomass doing this. It's basically like growing these cultures up in a bottle in the lab. You're doing it in the field. And because you have these high biomass, you can have high, fast kinetics, and you can really achieve low contaminant concentrations. The picture on the bottom left shows one of our cultures. It's the ENV425. It's a Rhodococcus ruber. It grows on propane, and it degrades a bunch of different contaminants. In this case, we did an experiment where we were degrading N-nitriso, we'll call it NDMA. And it was able to degrade, in just hours, the NDMA from 8,000 parts per trillion down to less than 2 parts per trillion consistently. And we've also been able to do this with the same culture and through biostimulation in the field scale via both bioreactor and biosparging approach, and achieved similar low parts per trillion levels. Also, one of the other advantages is that the co-metabolic organisms are widely distributed. You wouldn't think that these organisms that need oxygen to survive would be in anaerobic aquifers, but they are. And if you add oxygen and nutrients if necessary, in a matter of weeks, you can grow up pretty high populations of these organisms that are capable of degrading the contaminants of concern. And the final advantage here is that typically there's no toxic daughter products that are generated during this process. I give this example, TCE. Under anaerobic conditions, you would go through cis-DCE and vinyl chloride, which are not ideal places to be. But in this case, you go right to CO2. There are some intermediates, but they're not toxic, and they're typically degraded very quickly during this process. So these are some of the advantages of co-metabolism. So getting to the field demonstration, we performed this field demo at the former Myrtle Beach Air Force Base in South Carolina at the Building 324 plume. So this is located just off the Atlantic Ocean. You see on the bottom left picture, on the top right, you see our demonstration area is located about 1,000 feet west of the runway for what is now the Myrtle Beach International Airport. It was the former Air Force Base. So just a brief history on the site. Back in the 80s and 90s, they did a lot of remedial investigation. They found a rather large plume containing TCE, cis-TCE, and vinyl chloride above MCLs. In about 1994, that plume was measured about 2,000 feet long, 1,000 feet wide. And they installed a pump and treat system and ran that for about 11 years from 1995 to 2006. And then they hit asymptotic levels for treatment in the plume, and they shut down the pump and treat system and moved on to an M&A remedy that was approved by the regulators. Now, 15 years later, we still have concentrations within that plume, throughout the plume that are still, for cis-TCE and vinyl chloride, and in some spots TCE above the MCLs. But the M&A is at this point, showing that the plume is not expanding. So we did a lot of site characterization work for this project. I'm not going to get into the details, but we came up with a conceptual site model based on all this work that showed the plume in our area, which is near the toe of the plume, is about 210 feet wide and up to 15 feet thick. These are with cis-TCE and vinyl chloride concentrations above the MCLs are shown within the red dashed area. So we have a multi-level model here at the site. We have three units that are sitting on top of a clay. The clay is essentially an aquitard, so the contaminants reside in the two layers above that. Well, at first glance, these layers look pretty permeable, and this sand and shell hash layer, this dashed one here that's outlined in green, is very permeable when you look at it, and horizontal permeability based on HPT testing showed it was permeable as well. But when we did sparge testing at the site, we realized that it was very anisotropic, meaning that it had a high horizontal hydraulic conductivity, but a very low vertical hydraulic conductivity. And we determined that to be because if you could look at the picture on the bottom in the center, it has a lot of shell fragments in it. And these shell fragments are up to a few millimeters in size, and they were generally laid down flat in layers throughout this unit, and they act as a bedding feature and really prevent vertical distribution of the gases when we sparge out there. So that made us change our approach, which was originally supposed to be a horizontal well at the bottom that we would sparge gases up through this treatment zone to a different approach, which I'll talk about in a couple of slides. So just real brief background, we grabbed some soil and groundwater from the site and did a series of microcosm tests with different gases. We saw that ethene and propane were very good gases for treating the two contaminants of concern. And if you look at the top graph, which is vinyl chloride, you can see the concentrations in all these different treatments, some of them having propane, some of them just having oxygen, and some having different types of nutrients in there. We basically saw degradation of the vinyl chloride over time in all those treatments. We respiked a couple of those treatments. But in the ones where we added diammonium phosphate and the gaseous nitrogen amendment separately, those two treatments went down much faster. Well, if you look at the bottom graph, the cis-TCE was definitely, it's a different story. We only saw in the or this diammonium phosphate were the only ones where we got real treatment of cis-TCE. And we spiked it a couple times, after we stopped adding the gases, and we saw continued degradation for greater than a month in those treatments. One of the things we learned is that we needed to add a nitrogen source for this process to work at this site because of nutrient limitations within the aquifer. So getting back to how we were going to overcome this problem with the gases not distributing vertically very well was we had to go an approach where we installed wells at 3 different intervals. We know we were getting at least a 10-foot radius of influence, so we spaced the wells out at 20 feet at 3 different intervals for a total of 22 sparge wells to cover that treatment area. We used direct push to install these pre-packed well screens for the sparge wells. They went very quickly. We put in about 5 a day. And the picture on the bottom right shows what one of the well heads look like. That copper colored fitting there is a flashback arrestor that should there be a unlikely combustion within the well because of the gases we're using, it would prevent that from getting back to the system. We had several of them within the system as well as fail safes. The monitoring well network was extensive for this project. We had a total of 27 monitoring wells and 6 vapor probes. This cross section shows groundwater flowing, from left to right, basically going through our treatment zone. The green oval shows our 3 sparge wells at the different intervals. The red square shows basically our area of influence from those sparge wells. So we have wells within the treatment zone, but we also have several wells down gradient and wells above, and then wells up gradient of the treatment zone. These are our control wells. So for the system design, we used 4 different gases. We had compressed oxygen and nitrogen cylinders, as well as liquid propane and hydrous ammonia cylinders. And this system was solar powered and completely off the grid. We had 11 solar panels, a series of 12-volt batteries. We had remote access via a cellular system, so we could dial right into the system. We rarely needed people on site, except for oxygen changeouts. We could operate this system remotely from your computer, in my case, 600 miles away from the project site. The picture on the bottom left basically shows our gas distribution system. It's actually fairly simple. It's a series of solenoid valves and flow control valves and flow meters that control where the gases go, which gases go, and for how long. And that's all. We can access that remotely and change those conditions whenever we need to. So this is what, if you dialed into the system or if you were at the computer at the site, this is what you would see. It's basically a representative of that photograph I just showed of all the different solenoid valves and flows and pressures. So we operated for 9 weeks where we just had the oxygen. That was our control phase to see if we were doing any stripping of gases, and also to get the treatment zone aerobic. And then we did 12 months of co-metabolic biosparging where we had oxygen and also the two gases. Another thing that I should note is that we had 4 dissolved oxygen probes installed in monitoring wells within the biobarrier, and that data is presented right here. You can see the different concentrations of dissolved oxygen, and that let us know when we needed to sparge again, the oxygen to keep aerobic conditions. So it was nice. Again, that's totally remote. You didn't need to take field measurements. Everything is right here on the computer screen. So when we operated the system, we sparged 1 well at a time, starting with the bottom wells and working our way up. We were sparged at 10 to 12 CFM, and for about 5 minutes at each well. And we did oxygen 1 to 2 times per week, and propane and ammonia was sparged mixed with nitrogen approximately 2 to 4 times per month. So how much of the different gases did we use? Once we got going, we were going through about 20 pounds of oxygen per day. This was a very anaerobic aquifer, so it was kind of worst case scenario. So on an aerobic site or less anaerobic site, you would go through a lot less. Propane was about 45 pounds per month, or a half a cylinder. We had 6 cylinders that would lasted the whole demonstration. Ammonia was only about 1/8 of a cylinder that was needed. What I should note here is, I have this big X in the middle through this propane tank. We do not recommend that you use barbecue grade propane. That is full of other things besides propane, including propene and other compounds that can actually inhibit this process. So we use 99%, which is chemically pure grade propane and other gases for this process to work effectively. So getting into the data a little bit, like I said, we had 27 wells. Of those, 18 wells were within the treatment zone thickness and down gradient within that same thickness. So zones where the contaminants were above MCLs. So I'm kind of breaking these down into two groups. So the first 10 wells are located, between 5 and 10 feet down gradient of those sparge wells. Again, this shows the biobarrier being about 220 feet by wide by 20 feet long. And if we get into the data a little bit, so first talk about the cis-DCE concentrations at those 10 wells. There's a lot of data here, but I want to kind of break it down into 4 different phases. So as I mentioned, for 9 weeks, we sparged oxygen only. Then we added some propane, and we kind of got into a growth phase where we were waiting for that propane and that ammonia to be degraded. Then once we monitored the propane concentrations, we saw that being degraded. We started adding propane and ammonia on a regular basis every week to every 2 or 3 weeks during this treatment phase. And then there was we shut down, and then 3 months later we came back and did monitoring, after to see if we got rebound, which we expected because this is a barrier approach. And once we shut down treatment, we expected concentrations to come back. On all these figures that I'll be showing, the dashed lines are upgrading control wells, so they're pretty consistent throughout. But you see, we don't have much going on during the oxygen only phase or the growth phase. But once we start degrading propane and we get a high biomass out there, we see treatment of the cis-DCE. There's one well, this green well, which is we didn't see good distribution of gases at one particular well. And we also didn't see good degradation at that. But all these wells during this treatment period were below the MCL 70 parts per trillion for this particular contaminant. And then we did see rebound 3 months after we shut down. If you look at monochloride at those same wells, concentrations are lower, but they're still 5 to 10 times above the MCL of 2 ppb. And same story here, we see good degradation. Most of those wells are below our detection limit of 1 ppb during the treatment phase. And at the very end, we did learn that it's better to sparge more frequently at lower doses of the propane and ammonia than to do higher doses and longer periods in between. The interesting thing here is that the post-treatment numbers actually were higher than when we started. And at first we couldn't figure out why, but we looked at the background wells, this one that's shown in the black dash, as well as the other one. And those concentrations were increasing throughout the demonstration at both of those wells, which indicated that monochloride concentrations in this portion of the plume were actually coming up during this 15 months. So we were probably actually degrading more monochloride than we thought from the baseline conditions. So the next group of wells we're looking at is these 8 wells that are 15 to 25 foot down gradient. So these are outside of the influence basically of the sparging, but this is where the water that's being treated within biobarrier is moving through. So similar results here. We've got 8 wells in this zone. We've got degradation, good degradation at 7 of them. Again, we've got one outlier here. That's another well that we didn't get good distribution of the gases to. But again, all these concentrations were significantly reduced during the treatment phase and came back up at the post-treatment sampling event. And for monochloride at these same wells, we had good degradation kinetics. There was one well where we kind of flirted with the MCL of 2 ppb. I think it went below for one sample point, but it was generally above 2 ppb. So we'll say we had 7 of the 8 wells were below MCLs at these locations. So just real quick, looking at the ORP results. So this kind of gives you an indication of how anaerobic this aquifer was. The top chart shows the wells that are 5 feet down gradient. ORPs, started out baseline at about minus 100 or less, so pretty anaerobic. We started adding oxygen, and we got it oxidizing conditions out there, and it came right back. Within 3 months of shutdown, it was back down to minus 100 or more. If you look 20 feet down gradient, the graph on the bottom right, we didn't see much of an increase in the ORP at those wells, which were just, like I said, just 20 feet down gradient of the sparge wells after adding thousands of pounds of oxygen. So this aquifer is really what we're calling had a high oxygen buffering capacity, and that term was like coined by Hunter Anderson of AFCEC during my last IPR for this project, and I liked it, so I'm going to borrow it for this presentation. But basically, we had to consistently add oxygen to keep conditions within the biobarrier optimal, and also, as soon as we stop adding oxygen, it would drop very quickly. So we also collected some microbial samples from select wells during the demonstration. This is one of those wells. So we were looking at these various different co-metabolic genes. I think there were a total of 8 we looked at. These are the 4 where we saw increases. And what's kind of interesting here is the blue line is when we started adding oxygen, and we saw a big increase in these two sets of genes, these soluble methane monooxygenase and this oxyalkane transferase. And why did we see them before we started doing anything? Well, it's an anaerobic aquifer, so there was already methane out there. As soon as we added this oxygen, we saw an increase in the methanotroph population. And probably the reason we saw these other organisms increase is because they have the ability to grow on vinyl chloride as well as ethene. So with the vinyl chloride present and the addition of oxygen, we saw an increase in these oxyalkane transferase genes that are associated with these organisms. But we didn't see any increase in these other 2 monooxygenases until after we added the propane. So this is the propane monooxygenase and this short chain alkane monooxygenase or SCAM. And there's a lot of organisms that both these enzymes can use propane as a primary substrate, and they have the ability to degrade different contaminants. So these are really the organisms that were doing the work on this project. So in the big picture, this is these 18 wells. We took the average concentrations from those wells, and we took the baseline and the final sampling event. We had a 78% decrease in cis-DCE and a 92% decrease in vinyl chloride during the demonstration. So the conclusions from this work showed that we could achieve significant reductions of our target contaminants, both within and downgrading of this biobarrier. We achieved the MCLs at most of these locations. And this system really required minimum operation and maintenance. Like I said, I was running the system from my computer, a couple times a week. I check in on it, and run some cycles. But besides that, the only main thing we had to do on site was switch out oxygen cylinders using a forklift every several weeks for those 16 packs of oxygen that we had. Another good thing about this approach is there's no negative impacts on aquifer quality, such as you might get with an anaerobic approach. There's no mobilization of metals, and there's no generation of daughter products. And really, this technology, a lot of people think it's complicated, but it's really based on air sparging technology, which has been around for a long time. So it's really just adding some safety features to that approach. So some of the potential benefits for the DoD is this could be an alternative to a more expensive treatment approaches such as pump and treat. And we also showed that this is pretty much a sustainable, semi-passive and cost-effective technology that's completely off the grid. It would allow you to put this down in remote areas where you don't have electric. And not only can we degrade the CVOCs that we targeted here, but a whole bunch of other emerging contaminants as I have listed here. We've actually done some of these in the field successfully with this approach already. And it also has applicability to these mixed plumes, which is CVOCs plus, some of these emerging contaminants. And with that, I guess you can take any questions that you might have.
Great, thank you so much. We do have a bunch of questions that have come in. So let's start with this one that is from Terra Systems. And the question is as follows. Would your approach work for a site contaminated with CVOC, chlorinated ethynes, and 1,4-dioxane?
The answer is yes. We've actually done this at a site at Vandenberg Air Force Base where we were evaluating this for 1,4-dioxane treatment, and we also had low levels of various different CVOCs, including TCE. And one of the byproducts of degrading the 1,4-dioxane was that we got degradation of these CVOCs as well. Now, it would depend on which organisms you have out there. As I mentioned, you know, there's these different enzymes. You know, there's the SCAM, there's the PPO. So the SCAM enzyme tends to have a broader specificity towards a whole bunch of different compounds. So if you have those organisms present and you add the gases that would activate those enzymes, you have the ability to degrade that whole suite of contaminants. We also, on another ongoing project, we're looking at adding 2 different gases to stimulate different sets of organisms for a mixed plume. So this approach is applicable towards those contaminants that you asked about.
Great, thank you so much. This next question is from Shook, Hardy, and Bacon. Can you elaborate on any challenges you faced with the lower performing wells and distribution of gases? And if you did, what would you change in future field demonstrations or applications?
Very good questions. So as far as the distribution of the gases, the area of the plume where we didn't get the good distribution is where we only had one level of wells, because that's where we identified the exceedances of MCLs to be only in that lower level. I believe if we had a second level of sparge wells there, we've gotten better distribution of gases and better treatment at those wells. So that's definitely a lesson learned, I think, for this particular site. And then, I'm sorry, what was the second part of the question? Oh, what we changed as far as operating? You know, one of the things we were originally going to do on this project is we were going to use air instead of oxygen, but having the off the grid piece associated with air generation requires a lot of solar panels and a lot of batteries to operate a compressor. So we didn't have that upfront capital cost money in the budget. That's why we used the oxygen cylinders. But you know, I think within about a 2-year time period, you could pay off that extra capital costs and operate using air, and that would reduce the O&M cost of this, you know, to even less than what it is now. You know, adding air and these gases, whether they be individual or adding these like propane and these other gases below the LEL to the airstream, which we have done on other projects in the past.
Great, thank you. This next question is from Langan. Can you use alcohols as co-substrate?
I'm not a biologist, but what I do know is that for some of these organisms, different alcohols have the ability these organisms can grow on them, but the alcohols will not get those organisms to express the enzymes needed to degrade the contaminants. Because for instance, with isobutane gas, you get isobutanol is actually a byproduct of the degradation, and that's what these organisms can grow on. But without the isobutane, they don't express that enzyme. So you know, I would refer you to maybe Paul Hatzinger in my group, who's a microbiologist. If you had further questions on what alcohols might work, but we know the same thing for methanotrophs. You can grow them on methanol, but again, they won't express the enzymes that are required for the processes that we want, degrading these particular contaminants.
Thank you, Dave. This next question relates to in-situ propane concentrations following your field demo. Did you go back and measure what concentrations you were able to achieve specifically of propane? Sure.
So during the demonstration, after a sparge cycle, the concentrations would typically in the treatment zone would typically be in the hundreds of ppb to maybe a couple ppm range. We don't want to put in too much because because there's competitive inhibition. So that's something I didn't talk about because there wasn't much time. But if there's a lot of propane there, the organisms will prefer the propane over the contaminants. So you want, that's why we sparge in cycles. We sparge some propane and we let that degrade down to nothing basically. And those organisms are there, they're active, and all they've got left at that point is the contaminant. They don't have any propane. So you get really good degradation kinetics during that phase. So if you add propane too much or too frequently, you're not going to have good contaminant degradation because of the competitive inhibition. But basically, it's a matter of hours or a day or two, at the concentrations we were adding before all the propane is gone. So when we shut down the system, within a couple of days, all the propane was used up.
Great, thank you. This next question is from TU Dresden in Germany. You said that you were able to mineralize your target compounds and you produce as a result carbon dioxide. Did you see, or did you measure any impacts to groundwater quality, specifically to pH, following your successful demo?
Sure. We measured field parameters during all of our sampling events. So all 27 wells, we measured pH, ORP, dissolved oxygen, specific conductivity, and we did not have any change in pH that was noticed, during the 15 months of operation. And we also monitored for anions. We did see some changes in sulfate concentrations as we oxidized sulfide minerals. But there was an initial spike and then it dropped back down over time. We did not analyze for other metals. We did initially when we did site characterization, but there wasn't much in the way of dissolved iron or manganese out there. So we really didn't analyze. We knew those concentrations would only go down with addition of oxygen. So we didn't analyze that as part of the demonstration.
Great, thank you so much. This next question has to do with any unintended consequences. Do you experience any biofouling of the sparge well during your field demonstration? And if you did, how did you overcome that?
Okay, we did not experience any biofouling. So typically, if you do a groundwater recirculation approach, you're much more prone to biofouling than you are with a sparging approach. So with the sparging, so a couple things. Each of the sparge cycle was followed by a nitrogen purge. So we pushed all the co-metabolic gases and oxygen out of the sparge wells, so which would limit the growth in the well itself or the well screen. And I think that just the shearing forces of the sparging itself kind of prevents biomass from growing up on the screens. Part of this system, we were able to log all the pressures when we were sparging at each of the wells over time, and we did not see any increase in sparge pressures from the time we started the demonstration to when we finished at any of the sparge wells. We did not we didn't set a camera down or anything and evaluate the wells for biofouling, but the fact that we were still easily able to sparge in them without any pressure increases suggested that the biofouling was not an issue.
Great, thank you so much. And before we pull Shelly back into the discussion, just one last question for you. Did you sample for native microorganisms and cultures before starting your sparging demonstration?
We did, and that one slide I showed, that first data point was our baseline data point. We collected microbial data on, I think it was 8 different oxygenases, and those numbers were all very low at that first baseline event. And as I showed in that graph, they increased. Some of them increased when we added just oxygen, and then we had really big increases in the two oxygenases that we were interested in, which was the PPO and the SCAM, after we started adding the propane.
Great, thank you so much. At this point, we're gonna just try and wrap up with some general questions. we're going to go ahead and start with Shelly. Shelly, what are the next steps in terms of expanding your work and doing more field testing?
Yeah, thanks, Rula. I think we are continuing on both, I guess, sides of the project, if you will, on the science, on the fundamental, really trying to understand how exactly the microbe can degrade dioxane and chlorinated solvents and subsist under anaerobic, long anaerobic periods to really get to the fundamentals. And we'll be using omics-based tools for that, metabolomics, everything to confirm what we are seeing and the mechanisms. On the applied side, we are quantifying the rates, the kinetics, sensitivity, trying to go much lower than 10 micrograms per liter or even lower. The question is, how low can we go with respect to dioxane as a starting concentration, but how low can we achieve that? And then working in collaboration with Matrix New World and Geosyntech and the DoD, we'll be demonstrating ex situ and in situ application of this KB1 CB1190 mix, so that a single injection can take care of TCE and all its daughter products, and also dioxane without accumulating any toxic intermediates. So yeah, ex situ and in situ on both. And then we have some other creative things that we are trying in the lab.
Wonderful. Thank you so much, Shelly. Very exciting. What about you, Dave? Do you have any follow-on work that is being planned, or any field scale, full scale applications for the technology that you described today?
We do, actually. We have an ongoing ESTCP project looking at a mixed plume where concentrations are higher for CVOCs and 1,4-dioxane. And I mentioned that before. We're adding two different gases there to stimulate two different sets of organisms capable of degrading that mix of contaminants. On that project, we're doing a combination actually of groundwater recirculation and sparging. It's a little more complex. And then we have an upcoming project that's just getting funded. Paul Hatzinger has, that we're looking at acidophiles, methanotrophs that can exist at low pHs and degrade these contaminants in aquifers that have very low pH. And that's the same approach here, where we'll be adding probably methane and air as the sparging gases.
And Dave, when do you expect the results of your work, some of the stuff you described right now, when do you expect some of the results to become available?
So the mixed plume project with the multiple substrates is, we're actually building the system as we speak. So we'll be getting that started in a couple of weeks. That's a year-long demonstration. So I would expect we'd have some results in 6 or 7 months on that project. And the other project is actually just getting contracted, so it's a couple of years off before we would see results on the low pH project.
Great, thank you. Shelly, do you have any 30-second sound bites, last messages you'd like to leave with our audience today, or takeaway or take-home message before we wrap up, please?
I'm going to put my professor's hat and just say, the microbes, environmental microbes always come up with surprises for you. Things that you wouldn't expect. It's always a delight. Of course, you can't plan for this. In my time working on projects with just fantastic people in academia and industry, and I'm especially grateful for startup support to my research, including continuing support for this project that I described, that it just takes, the flow of not just samples and data, but even ideas and learning is bidirectional. I always thought you do stuff in the lab and then take it to the field. It was one way, but it is both ways. And that's very exciting to me as a scientist, applied scientist. So thank you.
Great. Well, I want to thank you both for an amazing job, and remind all our audience members that the audio and a copy of the presentation of today's session will be archived in our on our webinar webpage in case you'd like to refer to them in the future. Our next webinar is on Thursday, March 11, in 2 weeks, in the Resource Conservation and Resiliency Program area. This webinar will focus on new resource conservation paradigms on DoD land, and will feature 2 speakers, Dr. Dave Lytle from Oregon State University and Dr. Brian Hudgens from the Institute for Wildlife Studies. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other future webinars to the end of 2021. At this point, I'd like to remind you to please complete the very short survey that will pop up on your screen. This concludes today's webcast. Thank you to our speakers and to all of you for your attention. Bye-bye.